Welcome back, students. Welcome to biology classes. Today we are going to see 11 science biology and we are going to see the next few topics of chapter number 9, that is biomolecules. We have already covered so many topics of this chapter, and if you wish to check my previous videos of this chapter as well as other chapters, and you can check my YouTube channel for it. So without wasting any time, let's start with today's topic. So how do enzymes bring about such high rates of chemical conversions, right? So today we are going to see that how the enzymes are working or are bringing out the high rates, that is how they are increasing the rate of reaction, right? So the chemical or metabolic conversion refers to a reaction. So here what happens when an enzyme works, what happens, the chemical or the metabolic conversion, right? This two of the things occurs in the reaction. So as the metabolism takes place as well as the chemical reaction which is taking place, right, this is going to occur in the presence of the enzymes. The chemical which is converted into a product is known as substrate. So here, whichever chemical has been given, right, this particular chemical which is going to be converted into the product, this product which is formed, this is said to be the substrate, right? So when a conversion or a chemical or a metabolic conversion is taking place, right, a reaction is occurred, and when the reaction is occurring, at that time the chemical which is being converted into the product, that is known as the substrate. Therefore the enzymes having active site converts the substrate into the product. So here what happens, when the chemical is present, right, this chemical, right, this particular chemical gets converted, or the enzymes, right, the enzymes, they are having the active site, right? So this active site, the enzymes binds with the substrate, and when the enzyme binds with the substrate, right, it forms the product. So the substrate is being converted into the product. So the first main step which is been taking place over here is the binding of the enzyme occurs with the substrate. So none another, the substrate is being converted into the product. So substrate gets converted into the product, right? So now when the substrate binds through active site of the enzyme, ES complexes form. So once the substrate is binding with the enzyme, so the enzyme is containing the active site, and at this active site the substrate is able to bind. So when the substrate binds with the enzyme, right, it forms a complex which is known as enzyme substrate complex, right? Simply in short, it is known as ES complex. Now during the state where substrate is uh, bind to the enzyme, right, active site, a new structure of the substrate is called transition state. So here what happens, when the ES complex is formed, right, now when the substrate is binded with the enzyme's active site, what happens, thus there will be some changes which is observed in the structure of the substrate. So normally if a substrate is round in shape, right, after binding of this substrate or this round shaped substrate to the enzyme, when it takes place, the circle or the round shape of the substrate gets changed, right? So the structure of the substrate is changing, and this is known as this state is known as a transition state. Why it is known as transition state? Because there is some transitions which is taking place. Now the structure of the substrate gets transformed into the substrate of product, right? Now here what happens, whatever substrate, whatever is the structure of the substrate is, right, once it binds with the enzyme's active site, it gets transformed into the substrate of the product, that is the, it forms the changes, and when it forms the changes, right, that is said to be the product, right? Now all intermediate structural state are unstable. So unstable, that means they are not in their stable, they does not have the stability, right? So in the middle reactions which are taking place, right, so those middle reactions, they are said to be the intermediate reactions, right? So the all the intermediate structural states which are occurring in between the whole reaction, those all the states are in all the like states are unstable, right? That means there will be no stability which is observed. Stability is related to energy status of the molecule. Now stability in terms of that what exactly the energy status of that particular molecule is, when there will be the difference between the substrate and the product is observed, right? So the energy level difference, that is how much amount of substrate is needed to be converted into the product, it is based upon the amount of energy which is present. So the more, if more amount of energy is present, right, then more amount of substrate can be converted into the product, right? So it is based upon the energy status of a particular molecule. So based upon the energy status, the whole chemical reaction is taking place, and if proper amount of energy is there, then the stability is being stability is being observed, right? If product is at lower level, then substrate, and then the substrate reaction is an exothermic reaction, right? Exothermic, exo means outside, right? So here what happens, if a product is as it is at lower level, that means if the amount of product which is obtained is at lower level, right, then the reaction is said to be the exothermic reaction, right? So the whatever conversion of the substrate is occurred in the formation of the product, right? That means the conversion of the substrate to product is taking place. But which type of reaction is taking place? Exothermic, right? That is, there will be supply of the heat, right? And endothermic reaction, that means they does not need any energy, right? So there will be no supply of heat energy, right? So heat or either energy. So the reaction which needs energy, or right, which uses the energy, those type of reactions are known as exothermic reaction. And endothermic reaction means those reactions which does not need the energy, right? So in the endothermic reaction, the substrate has to go through a much higher energy state, and that is the transition state. So the reactions uh, which are going through very much higher energy state, those all reactions are said to be the endothermic reaction, and they reach a state which is known as transition state. Now there is also presence of the activation energy. So before that, you need to understand that what exactly activation energy is. So the difference in average energy content of substrate from that of transition state is called activation energy. So here what is that? That whatever difference, right, the difference in average energy content, right, that means how much amount of substrate is present and how much substrate it is utilized in order to reach the transition state, right? That means how much energy is needed, right, of the substrate to reach the transition state, that is known as activation energy. That means the energy which is utilized to reach the transition state, that is known as activation energy. So here you see, this is the reaction which is observed here. This is enzyme plus substrate, it forms the ES complex. Once it forms the ES complex, it is getting converted into a complex which is known as enzyme product complex, and this enzyme product complex get dissociated into enzyme plus product. So here free enzyme is produced and the product is released out. This free enzyme is again used over here, right? Next is, this is the graph which is shown, right? This is, you see here, if the particular, this is the potential energy. Now this is the substrate, that means how much substrate you are using here, how much energy, see this much energy is being used to reach at this stage. This is known as the activation energy, right? So here if it is reaching up to this, right, then this is in the presence of the enzyme, and here this graph shows is without the activate or without the enzyme. So if there is absence of enzyme, it needs more energy to reach this stage, but if there is presence of the enzyme, then in short time you can reach the activation or you reach the transition state, right? So this shows that in the presence of the enzyme, you easily get the product in very short time. But while we are talking about the reactions which are occurring in absence of the enzyme, so it takes a longer time to reach this transition state and forming the product, right? So this is about the two different types of situations which are seen over here. First is with the enzyme and second is without the enzyme. Next is, now what is the nature of the enzyme action? Normally, the whole uh, theory is based upon this reaction. This enzyme plus substrate, it forms the enzyme substrate complex. this enzyme substrate complex gets converted to enzyme product complex, and this enzyme product complex, it gets dissociated to form the free enzyme plus the product. So when enzymes combines with the substrate, it forms the enzyme substrate complex. So just I said over here that enzyme plus substrate, it forms the ES complex, and that is enzyme substrate complex. Further, this enzyme substrate complex, it dissociates into enzyme product complex, right? So here ES gets dissociated to form the EP, which is known as enzyme product complex, right? Next is, upon dissociation of this in EP complex, right, a free enzyme, that is E, that is a free enzyme plus the product, right? This is formed when this EP is dissociating, right? So this is about the enzyme action which is taking place. So you need to write this 3 steps. The first step is the combination of enzyme and substrate, and upon combination they form ES complex, that is enzyme substrate complex. Once this enzyme substrate complex is formed, it dissociates to form the EP complex, that is enzyme product complex. This is the step second. And the third step is the dissociation of this EP complex into the free enzyme and the product. Next topic is the factors which are affecting the enzyme activity. Now there are several factors which are affecting the enzyme activity. That is, it can reduce the amount, or it reduces the activity of the enzyme. So enzyme activity is affected by the change in conditions which alters the tertiary structure of protein. Now here the enzyme activity is affected by several factors in different conditions, right? So based upon the type or the change in condition, right, it changes the tertiary structure of the protein. And when the changes of the protein takes place, what happens? The enzymes are not able to work properly. So the factors which are affecting the enzyme activity are pH, temperature, the change in substrate concentration, right? So we are going to see the 3 of them in detail. First we will see about the temperature and the pH. So the enzymes show its highest activity at particular temperature and pH, which is called as optimum temperature and optimum pH. Now, see the temperature or the pH at which it shows highest activity, right? If it shows at the highest temperature, highest activity at a several temperature, that temperature is said to be the optimum temperature. Same way, if an enzyme is showing highest activity at a particular pH, then it is known as optimum pH, right? Now at low temperatures, the enzyme goes in inactive state, right? So whenever the low temperatures are present, right, the enzymes, they are going in the inactive state. But when the higher temperatures are present, at that time the enzyme activity is destroyed, right? Why it is destroying? Because the protein gets denatured at higher temperatures, right? So at lower temperatures, it goes in the inactive stage where it cannot work properly and the reaction cannot take place. And when the temperatures are comparatively high, at that time the protein is denatured. So again the enzyme cannot work properly. So this is about the first, that is the temperature and pH. Next is the concentration of the substrate. When the substrate concentration increases, the velocity of enzymatic reaction rises and reaches the maximum velocity, that is Vmax. Now, when the concentration of substrate, that means if the concentration of the substrate is increases, that means if the substrate is present in more amount, then the reaction is also increasing and it reaches a maximum speed or a maximum velocity, which is known as Vmax, right? So Vmax is what? That is the enzymatic action or the enzymatic reaction. It increases and reaches the maximum velocity level, which is known as the Vmax, right? The enzyme activity is sensitive to the presence of specific chemical that bind to the enzyme. Now, there should be no presence of any of these specific chemical, because these chemicals can easily bind with this enzyme. And if now here what is needed to be bind? The enzyme should always bind with the substrate in order to get the proper amount of product. But if there is presence of any of the specific chemical, then what happens? This chemical has the tendency or has the ability to bind with this enzyme. So rather, if the particular substrate is about to bind with the enzyme, but what happens, this chemical binds to it. For this reason the substrate cannot bind with the enzyme, and for this reason what happens, the chemical reaction is not taking place. When chemical binds to the enzyme and stop the enzyme activity, the process is known as inhibition, and the chemical is said to be the inhibitor. So here what happens, the chemical which is binding with that enzyme and it is stopping the enzyme activity, process is known as inhibition, that is stopping the process, right? And as it is responsible for stopping the process, it is known as inhibitor, right? So the process is known as inhibition and the chemical is said to be the inhibitor. Now first we will see about the competitive inhibitor. Competitive, that is some competition which is taking place. That means here the inhibitor, that is the chemical, is almost very similar to the substrate. That is the shape and the structure is same as the substrate. So now what happens, the enzyme, instead of binding with the substrate, it binds with the inhibitor. And as it binds with the inhibitor, what happens, the particular reaction is stopped, and this way the competition is taking place between the inhibitor as well as the substrate. So this is the graph of the three of the factors. This is the effect of change in pH. This peak level is said to be the optimum pH. Same way here, this is about the temperature which is seen in the like change in temperature, but as it is reaching over here, this peak level is said to be the optimum temperature. And the third is the concentration of substrate on enzyme activity. See, you can see here, this is the substrate level, right? And this is the velocity, right? So they are utilizing the substrate and they are utilizing the substrate and they are reaching a maximum velocity which is known as V max. And the half of it is said to be the V max by 2, that is half of this velocity, right? So this is about the factors which are affecting the particular enzyme activity. Now we are going to see about the classification and the nomenclature of enzymes. Now we are going to see the 6 different types of the classes of enzymes, right? So six different classes are there which are going to perform different types of functions, right? First class is the oxidoreductase or dehydrogenase, right? So these enzymes which catalyze oxidoreduction between two substrates. So here the first type of class, which is the oxidoreductase or dehydrogenase enzymes, right? They carry out the oxidation reduction reactions. And when the oxidation reduction reactions are carried out, right, between the two substrates, right? So there are so many different types of enzymes which are present in this class, which are doing the functioning or which are doing the oxidation reduction reaction, right? So whichever enzymes which are responsible for carrying out the oxidation reduction reactions, they all are placed in the first class, which is known as oxidoreductase. Second is transferases. Transferases, that means the name itself depicts that it is going to do the transfer of a particular group. So enzyme catalyzing a transfer of a group. That means whichever group is being transferred from one place to another place, this type of enzymes are used. So sometimes these enzymes are responsible for transferring a particular group from one molecule to the next molecule. This type of enzymes are placed into the second class, which is known as transferases. The third class is the hydrolases. Hydro means water, lases means the hydrolysis, that is the breakdown of the water molecules, right? So the enzymes which are going to carry out the hydrolysis of the ester bond, peptide bond and glycosidic bond, this three of the bond is being broken down with the help of specialized enzymes which are going to do the hydrolysis, that is the breakdown of the bonds which is present in between the biomolecules, right? So those type of enzymes are placed into the third class, that is the hydrolases. Fourth class is the lyases, and these enzymes are going to catalyze or they are going to work on the removal of the groups from substrate. So it is this lyases, they are going to remove several groups from the substrate which are not needed. So all the enzymes which are going to remove the groups from the substrate, that those all are enzymes are placed into the fourth class, which is known as lyases. The fifth class is isomerases, and this isomerases includes the enzyme catalyzing interconversion of isomers. Isomers, that is they are similar to each other, right? Nearby similar to each other. So this type of enzymes, they are going to interconversation, right? That is, they are going for the changes, they are going to observe the changes which are formed in between the isomers, right? So all the enzymes are included in isomerases. And the last class is ligases, and this ligases, they are responsible for joining the two compounds, right? That is, if a particular one molecule is needed to bind with another molecule, this type of enzymes are being used. So these are the 6 different types of classes of the enzymes, right? Now we are going to see the last topic of this chapter, and this can be asked in 2 marks, right? Next, last most topic is the cofactors. So it is a non-protein chemical that assists with a biological chemical reaction. See, this is a non-protein chemical. That means there will be no presence of the protein, and this is going to carry out the biological chemical reaction. So it is not protein, but still they are working in the human body, as well as they are working in the they are going for the product reaction which is going to take place. Now in this you are going to see about the prosthetic group, coenzymes, and the metal ions, right? So the prosthetic group are organic compounds which are tightly bound to apoenzyme, right? Apoenzyme is nothing but the inactive enzyme. So this prosthetic group, they are organic compounds and they are very tightly bound with the inactive enzymes, right? Seconds are the coenzymes. These coenzymes are organic compounds which has the transient association with the enzyme. So this coenzyme that is working together, right? So this organic compound, they help the enzyme to carry out the activity of the enzyme. That is, it helps the enzyme to perform its activity. And the third one is the metal ions. And this metal ions, they are required for the enzyme activity. That is why they are responsible, why they are required, because they form the coordination bond with the side chains at active site and with the substrate. So it is this metal ions, they provide the proper binding of the substrate to the active site, as well as it also allow the side chains at the active site to bind properly with the substrate molecule. So for the proper binding of the side chain of the active site and the substrate, the metal ions are required. So these are the 3 different types of cofactors which are observed and which are present in order to increase the activity of the enzyme. So here we complete this chapter, and I hope you are very clear with this chapter, and if you have any doubts, you can check my previous videos of this chapter. So I hope you are very clear with today's topic, and I hope to see you again. Thank you.